Tate, who is in charge of NARM's program as a science leader at FDA. Dr. Tate has been studying and researching AMR surveillance system for 15 years, and she has been in charge of transatlantic task force as well as Codex ad hoc. She has been in charge of national task force as well as committee groups on behalf of the US government, and the topic of her talk is NARM's use of technologies for AMR surveillance and monitoring food. And she has been conducting various antimicrobial related research. Once again, the title of her talk is NARM's use of technology for AMR surveillance and monitoring in food. Please give her a big round of applause.
First, I'd like to thank the organizers for inviting me to talk about NARMs and how we're using these various technologies that I will be speaking on. I also want to say a disclaimer that these are my views and not necessarily the views of FDA, so just be mindful of that. All right, and so in the presentation, I'm going to give a brief overview of NARMs and how we're One Health monitoring. I'll talk about our use of whole genome sequencing, visualization tools that we've created, as well as the use of long-read sequencing, metagenomics, and artificial intelligence / machine learning. So NARMS is and has been for most of the existence of the program a tri-agency program. And so, we are a collaboration among 3 major government agencies. CDC is gathering data from sick persons, and that's coming from all 50 state labs. FDA, we are working with 24 state public health and university labs to go to their local grocery stores and collect retail salmon, shrimp, tilapia, chicken, turkey, pork, and beef, and they are isolating antimicrobial resistant enteric bacteria from those retail packages. And then USDA, or the US Department of Agriculture, they are in the majority of processing plants in the US, and they are collecting animal ceca as well as product testing from broilers, turkey, swine, cattle, and other animals as necessary for certain pilot studies that we might do. And that actually does not include shrimp. I'm just noticing that now shrimp, fish, and fish are not part of USDA's purview except for catfish. But all of these data are integrated into annual reports that we publish online. And then, we're expanding into the One Health paradigm. And so by doing that, we're also looking at animal health. And so we have newer collaborations with programs at FDA and the USDA that are collecting isolates from sick animals, both food animals and companion animals. And those isolates are coming from veterinary diagnostic labs across the US and even in Canada. And so most recently, we've included some of that data into our reporting tools. The other aspect of us reaching into One Health is that we have a pilot study looking at AMR in water. And so we are working with the U.S. Environmental Protection Agency. We're leveraging 2 of their existing surveillance programs where they're looking at, one is looking at national rivers and streams across the U.S. And so we've co-opted that, and we are looking at AMR bacteria and as well as antimicrobial resistant genes just in those national rivers and streams. And then there's also another pilot study that's on a smaller scale. It's in one watershed, but it's a very intensive pilot study in that we're collecting a lot of metadata from that study so that we can figure out what the drivers are for resistance in the water environment. And I know that, a lot of programs that are reaching into the environment now for AMR monitoring, they're looking at wastewater. We're starting with surface water, because surface water receives human, agriculture, and wildlife input, and they're also the source, water sources for humans, agriculture, and wildlife. And so the methods that NARMS uses involve culture-based methodology. And so, we're isolating our bacteria from a sample, and we're doing whole genome sequencing, and we're doing broth microdilution antimicrobial susceptibility testing. I should say that whole genome sequencing, we were actually able to jump onto that fairly quickly, and the reason is because we leveraged existing programs that the US has, such as PulseNet, which is at CDC, and Genome Tracker, which is at FDA, which has for many years used molecular techniques to identify and track outbreaks, foodborne outbreaks. And as they started to use whole genome sequencing, we were able to leverage those systems and use them for NARMS retail meat surveillance. And so really what NARMS had to do then was try to figure out how the whole genome sequencing data relates to the susceptibility testing data that we had been generating for years. And so we put out publications almost 10 years ago now, where we looked at the resistant genotypes and phenotypes across all the bacteria that we're testing in NARMS, which includes E. coli, Salmonella, Campylobacter, and Enterococcus. And we found that there was a really high correlation between the phenotype and the genotype for all of our NARMS organisms. Although it did somewhat vary depending on the drug that you looked at. And so, for streptomycin is an example of that, where we actually recommended that the breakpoint that we had generated, we called it a NARMS breakpoint. The breakpoint that we were using for our surveillance, we shifted it lower. And actually it's an ECOF because this is not based on PKPD data. So we shifted it lower because when we compared the phenotype and genotype, we got a better sensitivity and specificity when we lowered the breakpoint to 32 micrograms per mil. So the implementation of whole genome sequencing in our retail meat surveillance program has been stepwise. Since 2015, we have sequenced all of our Salmonella and Campylobacter, and since 2019, we've sequenced all of our E. coli. Prior to that, we were sequencing just multi-drug resistant E. coli, or sometimes every other E. coli. It varied by year. And then we're also sequencing select Enterococcus. And again, leveraging that partnership that we had with Genome Tracker Labs, we were able to send our historical isolates that we collected since the inception of our program, which began, the retail meat sampling began in 2002. So we were able to send all of those historical isolates to Genome Tracker Labs, and they did the sequencing. So we have all of our collection from 2002 to now 2024 has been sequenced. In 2019, we transitioned our sequencing responsibilities to the NARMS retail meat sites. And so now we have WGS data that's generated from the sites that is being directly submitted to the National Center for Biotechnology Information, or NCBI. And they're submitted under NARMS Bio Projects. And these data are shared with the public in real time. And because we're using whole genome sequencing, there's a greater sensitivity now in our surveillance for detection of emerging resistance and also more rapid detection of outbreaks. Beginning with 2023 retail meat data, we are sequencing all the Salmonella, Campylobacter, and E. coli isolates. but we're only conducting phenotypic susceptibility testing on 20% of those isolates. So that just speaks to our confidence in the whole genome sequencing data. Again, this is for surveillance purposes. I know we talked about, for clinical diagnostics, it might not be that reliable right now, but for surveillance, we have a lot of confidence in this so that we only have to now do susceptibility testing on 20%. And the reason that we're continuing to do it on 20% is because we want to, over time, see if the correlations that I talked about earlier, if those are stable. And then also, with whole genome sequencing, the fallacy of that is that you only know what you know. So if a resistance gene has not been annotated, or previously identified, then when we run our sequences against that database, it's not going to be identified in our sequences. So if we're continuing to do susceptibility testing, we might be able to uncover new mechanisms that might be emerging. So again, the states are submitting directly to NCBI their pathogen detection pipeline. Is what NCBI uses. So basically, the NCBI is clustering these genome assemblies, and then they're inferring trees from those clusters. But at the same time, they're also annotating the sequence data using tools like SeqZero to uncover the Salmonella cerevisiae, if it's Salmonella, or AMR Finder Plus, which identifies the antimicrobial resistance gene. The AMR Finder Plus also identifies virulence genes and stress genes. And so on the right, what you see is basically the output of the pathogen detection pipeline. And so we take all of that data, including user input metadata, and we put it into a visual analytics software platform called Tableau, which is a business analytics platform, but we're using it for our whole genome, to display our whole genome sequencing data. And the tools I'll talk about in the next couple slides are NARMS Now, Integrated Data, and Resistome Tracker. So NARMS Now, Integrated Data integrates resistance trends across all of the bacteria that we are analyzing in NARMS and across all of the sources that we are analyzing in NARMS. And the really nice thing about this tool is that it seamlessly integrates both the susceptibility